So now we will see that biochemical evaluations are biochemically how the evolution of the virus has become originated. We are just concerned with the early forms of the life, whether the early forms of the life will have basic structure. We have the similar kind of the structure with a different organism, just like we can assume that we contain the nucleic acids, or we contain the RNA, or we contain the DNA, or the similar kind of the basic constituents or basic structure was there. However, the most important is that how the replication was coming on, or how the synthesis or the metabolism or the reproduction and the utilization of the UV light suggested that the life was self-replicating. It is assumed that life was self-replicating and there was non-enzymatic synthesis. There was no need of the enzymes, and there was only something was present on the earth was the oxygen and the utilization of the UV light which was there. Then the evolution dependent on the reproduction of the external supply of organic building blocks. Organic building blocks are very important constituents of the life, so it was dependent on the reproduction on the external supply. And then more synthetic power by the mutation is also very important, that more synthetic supply by the mutations. And then selectively favoration for the naturally is very important for the early forms of the life. Then the appearance, how the early forms of life appeared. If we have the biochemical evolution of the viruses, biochemical differentiation depends on the synthesis patterns and the macromolecule productions, and then the chemistry and the metabolism. We already seen that maybe there was a non-enzyme, no enzymes were needed, and there was only the UV lights. But the chemistry and metabolism are again very important, and how the life was appeared and how the viruses can be originated without these things, biochemical. Here is an example of the replicase gene, that how the genes were carried on, and the non-viral replicons are there, and then the viral replicons. Here we can see just here the replicase gene is there, and the non-viral replicans and viral replicans capsid 3. We have used the 3 capsids, capsid 1, 2 and 3, and then it gives us the virions, different types of the virions, virion type 1, virion type 2 and virion type 3. 3 different types of the virions give us through the 3 different viruses, the capsids. And then the replicase type 1 and type 1 again the enzymes. Then the replicase type 1 give us the virion type 1, replicase type 2, and then replicase type 2. This is just the hypothesis of the evolution. This was just the evolution of the biochemical synthesis of the viruses.